everybody. Welcome to Skype a Scientist Live. My name is Sarah McNulty. I run Skype a Scientist, and today we are talking all about antibiotic resistance and specifically how we can look for new antibiotics in our environment. And so we are joined by the fantastic Nichole Broderick, Dr. Nichole Broderick. She is a professor at Johns Hopkins and was also on my committee when I was in graduate school at University of Connecticut. And she's also on our board of directors at Skype a Scientist. So lots of reasons Nichole is fantastic. This is all part of Antibiotic Resistance Week, which is a nationally celebrated week of awareness to be more aware of how antibiotics are awesome, but also how we have to be super careful about using them so that we don't accidentally create bacteria that can handle whatever we throw at them. And so Nichole will be talking more about that today. So Nichole, thank you so much for joining us.
Thank you so much for having me. I'm really excited to be here and talk to you about a very important topic and tell you about stuff that I do with students and a big community of scientists to try to understand this issue and come up with some solutions. So I have a few slides that I wanted to present to you. Hopefully that is coming up properly on presenter mode.
Looks good. Awesome.
So I'm here as part of World Antibiotic Awareness Week. It's an event that is it's recognized by both the World Health Organization, but in the United States, both the CDC, NIH, and then a lot of other international versions of things like the disease control are also really important for participating in this and doing local awareness campaigns, but then also things that we can do to understand both the issues better and be smart about how we use antibiotics and also just more aware of what the problems are. I'm part of a group called Tiny Earth, as I mentioned, and it's a course that was started to harness students in high schools, in colleges, and research institutions, and community colleges, all through the gamut, to really try to tackle this problem. And I'll explain more what we mean by that. So why this is such a pressing issue is because we have, and you probably hear about this in the news sometimes, or maybe you've talked about it in different science classes, or maybe you even had a friend or a family member who's, or you yourself have had one of these antibiotic-resistant infections. And that's that we see this rise in the number of bacteria and microbes, we might hear them called pathogens too, that are increasingly resistant to the antibiotics that we have to combat them. Our discovery of the production of antibiotics and the ability to use microbes in our world to be able to take this resource was a huge improvement upon our health. And it was very important for not only human health, but we use antibiotics in a lot of settings, whether it's for food production, like for taking care of our animals and making sure they don't get infections, or even our crops. We sometimes use them because there can be bacterial pathogens or fungal pathogens that can be damaging to them. The problem is that while we've seen this increasing rise in the number of antibiotic-resistant microbes, we have fewer and fewer new antibiotics that are being identified and that companies and pharmaceutical companies are putting into production. It can be really expensive to go find them and to do all the work to be able to get those to be able to be used in humans or other settings. We have to do a lot of trials to make sure that they don't have harmful effects. And a lot of pharmaceutical companies have done a lot of focus on other important problems. So one of the big ones, for example, is something like COVID, right? So we have this pandemic with this viral infection. And so that's been something where there's been such a massive need that that's been a big focus. Our antibiotic resistant infections aren't going away, but there's in some cases a little bit less incentive for companies because they don't make a lot of money on them. And unfortunately, a lot of these things can come down to that. But that's where we think students can come in and actually work to help solve this problem. And so the resistant crisis that we have is that we have these few new antibiotics, but more microbes that are resistant to it. So what is the solution? And I'm going to tell you about a few of them. And one of them in particular, I'm going to talk to you about Tiny Earth, which is near and dear to my heart. And the idea when this was first developed by our founder, Dr. Jo Handelsman, was she wanted to, first of all, have a course that would give students real research experiences. It wouldn't be just an experiment where we knew what the outcome was going to be. It would be actual true research that could be done by students who maybe weren't even in college going to be majors in science and or math, but just that were interested, and this would be a great way to expose them. And we also know from studies that the sooner students actually get experience with research, the more likely they are to actually take those additional courses. So it's an exciting thing. It's reasons why I became a scientist. This ability to discover new things and do something important that has value. And we want to give that experience to students. And so this course was started and has grown since 2012. It now includes about 700 instructors worldwide. And we figure that about 10,000 students, more than 10,000 students a year are doing this course. So if you think about that, of that number of people out there looking for new antibiotics, we think that there's a big possibility we'll find something new. We have this in about 27 different countries. We're increasing that number all the time, and 47 US states and territories. And so this is why what we do. So we focus primarily on soil, and there's a number of reasons. One is that actually most of the antibiotics that we commercially use now, so the tetracyclines and things like that, are derived from microbes, but they're also derived primarily from microbes from soil. And this is, we think, because soil is an extremely rich environment for microbes. There's anywhere from about 10 to the 11th soil bacteria per gram of soil, and there can be anywhere from 5 to 30,000 species. So it's an extremely diverse and abundant source of microbes. It's also something that's right out your back door, right? So I just went out in the backyard. It turns out to be a beautiful day here in Baltimore. I took advantage of it, just went out, got some soil from my garden. We're really tied to the soil where we live, and soils are different across the world, and they can have different properties that can affect the types of microbes there and the types of things they use. So one of the beauties about soil is that it's easy to obtain. We know a lot of good microbes come from it, and microbes that produce antibiotics. And so this is showing students out collecting, the pointer, students out collecting soil. We then bring it into the lab, the soil, and we, if you think about it, 10 to the 11th, that's a really big number. So 10 to the 9th is a billion. So this is even bigger than that. So almost a trillion bacteria. It can be a lot in there, but even think about it at a million or a billion cells. That's a lot of bacteria. So how do we get them from the soil? What we do is we use special food for microbes in the lab. This is a Petri dish. You may have used one of these in your science courses or just seen them on TV or things like that. And it has nutrients that allow microbes to grow. A lot of the same things that we need: sugar, amino acids, those sorts of salts, things that we need for growing, and especially water, because they also need water to be able to grow. And so basically what we do, so I set these up, they've settled a bit. I don't know if you can see them. Maybe I can use my fancy dancy home. I'm not in the lab because of the pandemic. So I'm having to make do with some home things, but this gives you a sense. So I'm using a syringe actually that I have an extra of that we use for giving my little daughter Tylenol, but it has measurements on it, so we can do that because we want to keep that. And so what I did there is I've made a dilution series, we call it, going from most concentrated to least concentrated. And so what we're trying to do is get those microbes into solution so that we can get them separated from each other, separated from the soil particles. And then we put them onto this media, and that allows them to grow. And we spread out that liquid so that we get the cells separated from each other. And ideally, you get to a density of cells where you have one cell that then starts dividing. And as it divides, it becomes a macroscopic, what we call a colony. And so here you can see some that are not separated, like they're growing into each other. But here we have some nice ones where we can actually see separation. And so what our students then do is they take those different microbes and they patch them onto other plates, and then they test them against other strains of bacteria to look for antibiotic production. And here's a beautiful example of that. So this is a tester bacteria. So this is a bacteria that we want to see if it is sensitive to antibiotics. And this is a microbe that might be an antibiotic producer. And indeed, we have two here that are really robust producers, I would say. One here that is also producing some. Here you can see one that doesn't seem to be producing any antibiotic that's active against this tester strain. It might be producing an antibiotic that another microbe is sensitive to, but at least in this case, this one that's being tested, it's not. And then we do other things to try to understand who those microbes are, what are they doing, and work on to other aspects to be able to isolate the chemicals that they're producing, that they're secreting into the media that causes this production. And I think I have a nicer picture of that here. So here you can see we have some good producers. Here's an array of different microbes that we want to be able to test. You can see all the beautiful colors these microbes have. Microbes don't necessarily, like if you think about a tree versus an elephant, they're maybe not necessarily that different. They're usually kind of either like what we call bacillus-shaped, rod-shaped, or circles, spheres. But microbes have excelled in metabolite diversity. So they don't necessarily look physically really different, but they produce all sorts of fascinating things. And you can see that here with just looking at these pigments. And sometimes pigments are actually antibiotics. So this is one reason why a lot of students really, just because they're pretty, like to take a lot of pigmented microbes. We then try to understand, isolate this specific one, and then we can do things where we use different solvents to be able to extract out those chemicals away from the bacteria and see if we have the active producer. And we do a lot of other characterization. And this is really valuable because it starts to let us then be able to take the chemical part of it and ask, is this something that's the same or different? And so our courses in microbiology and biology are typically doing this first major step. Some of our courses are starting to do things with the chemistry and even the genomics, so they can try to understand what genes might be responsible for producing these antibiotics. But we also have a chemistry hub that's based at the Wisconsin Institute for Discovery. And that's where classes and students can send in their isolates. And we have a team of scientists there who work on trying to understand these better. Because the goal is that we'll start to understand, the different genes that might be producing these, the different metabolites they might be producing, and ideally getting to a point where we can identify a new antibiotic. And like I said, we think, so one reason that a lot of pharmaceutical companies have stopped this is that when they go out and sample, they find a lot of the same things again. And so they don't want to put a lot of money into finding tetracycline 100 times, like every day. And so what we think, though, is that if you have 10,000 students doing this or more every semester or every year, the idea is that, yes, some of them will find tetracycline again. And actually, we want them to, because that means our methods work, right? That's a good validation that our methods are working. But more importantly, with that many hands at the, doing it, the student sourcing or crowdsourcing that we're doing, we think that we're going to be coming up with some new antibiotics, and we actually have some that are in analysis, and that we think we've identified at least some new derivatives or new types. So this is sort of the exciting thing that we do and our students are working on. I don't know, Sarah, if you want me to stop and take a few questions.
Let's do some questions. One thing that I'd like to gain some clarification on. So can you go back to the slide where you had those zones of inhibition?
Absolutely. Let's do this one, because I really like this one.
That's perfect. That's great. So just to clarify, so like what we're seeing here, each of these grids, you have a different type of bacteria. And then the like major kind of gray white that we're seeing kind of all around, with the exception of right around those two, that's the bacteria that we're basically trying to kill, right?
Yep.
And so the way we know if a bacteria that we collected from the wild is going to be maybe creating an antibiotic is if we see like a lighter, clearer looking jelly instead of the gray swath.
Yep. So this is called what we call a zone of inhibition. And so in this case, we're using, so the strains of bacteria that we like to use are ones that we know are problematic, but we use safe relatives. So we don't want someone using a multi-resistant microbe. We want them instead using a safe relative. So for example, you may have heard of MRSA, which stands for methicillin-resistant Staph aureus or Staphylococcus aureus. So Staphylococcus aureus is the type of bacteria. And it's been a problem because it has developed resistance to a number of antibiotics. And so we're really, this is one of the bacteria that we're really interested in finding new antibiotics against because of this. And so we wouldn't use Staph aureus in our classrooms, but we use like a Staphylococcus epidermidis, which, if you can understand the epidermis part, it actually is a microbe that lives in our skin, but they're related enough that the types of cellular functions that an antibiotic would target allows us to use that. So we can use a safe one. And so then that's what you'll see. So your student will spread that one down, and then they'll patch these potential producers. So these are the ones that they've collected from their soil. And what they're looking for are these, what we call zones of inhibition. And so this means that the antibiotic, the bacteria isolate from the soil, as it was growing, started secreting this metabolite, this antibiotic. And as the Staphylococcus was growing next to it, it was getting killed. So the one thing about how antibiotics work is they're being produced by a microbe as it's growing, and microbes are most sensitive to the antibiotic while they're growing too.
Awesome. So we have a question here from Patricia. So aren't antibiotics produced by fungi?
They can be produced by fungi as well. And the first one we ever the first one that was discovered and put into production was from, this is the classic case of streptomycin. And it definitely was from, I'm going to make a mistake, and it wasn't streptomycin. Oh my gosh. No, it was penicillin. Thank you. And I'm not teaching this semester, so I'm a little bit but it's totally penicillin. It's penicillin, yeah. And that was produced by a fungi. And so yes, antibiotics are also produced by fungi. They're produced by a lot of different types of microbes. Most of the ones we use, though, are actually produced by bacteria. We do have some classrooms that are starting to get interested in working with fungi to see about their production. But one of the fungi are a little more difficult to work with in some cases. And so we think for most classrooms, it's easier to work with the bacteria. But definitely microbes like fungi are another great source of antibiotics.
Awesome, thanks. The next question is from Amara. How fast do bacteria tend to grow?
It's really dependent on a number of things, like their own growth rate, which can be dictated by just their physical timing, but it also can be affected by their nutrients and what they're exposed to. So something like E. coli in the lab that we study a lot, it has what we call a doubling time. So that means it divides into one cell into two cells about every 20 minutes. There are some bacteria that can be even faster than that, 15 minutes or so. Then there's some bacteria, and some that you may have heard of as being problematic pathogens, like Mycobacterium tuberculosis, which actually take days to divide. So they're really slow. They can be very slow growing, and whether a microbe has a lot of access to nutrients can also affect that. So some bacteria, if they don't have enough food, and this actually happens a lot in the soil, they'll go into basically a dormant state where they won't be growing at all. And then when the nutrients become available, then they suddenly, if a worm dies nearby, then that maybe allows them to access that. So it can be really dependent. And actually, when we do these assays, that's a great question, because when we do these assays where you're challenging these two microbes technically to grow, against each other, if one of them grows too fast, you might not be able to see the effect of the antibiotic. So if the tester strain goes really, really fast, but the producer that you found from the soil grows slow, you might not be able to, it might still be producing antibiotic, but you just can't see it. And so those are some things that actually some of our classrooms have been really interested in testing. Mm-hmm. And so they've been doing experiments to try to alter, like, if you patch them a few days beforehand, and then you add the different tester strains. But it really is an important, aspect to get right as to how they're growing. So that's a great question to think about.
Awesome. And an assay is basically just like a type of experiment.
Yes, thank you. Yes.
Okay, so in your opinion, this is from Dean, what is the worst bacteria?
In my opinion, what is the worst bacteria? You mean in terms of like its impact on human health?
Let's give that as the definition.
Or do I have one that I don't like because it's difficult to work with in the lab? No, I have a personal issue with this bacteria. There's a lot of really, I think there's a lot of really, I think for my opinion, the worst bacteria that we have to be afraid of are the ones that are developing resistance to multiple antibiotics. Not just a single, but like almost, we have some bacteria that can be resistant to like all but some of our last line of antibiotics that we don't give routinely to people because we have to keep them in reserve for being able to do that. I think those are the scariest infections because, before Alexander Fleming discovered penicillin, and we were able to use that for human production and for improving human health. Something as small as getting a cut in your garden could lead to death, and did. And so, it's the scary thing to think about is that we could be returning to something like that if we don't find new antibiotics. And there's some predictions that by 2050, deaths worldwide due to antibiotic resistance will be the highest, much higher than things like cancer and stuff that we worry about all the time. So I think to me, that's the worst bacteria and the ones we have to think about and consider and think about our practices and what we're doing and also what we're trying to discover.
Totally. Okay, so quick question. Do you have any more slides that you'd like to show us? Or, and if not, we should minimize it because it'll make Erin larger.
Exactly, so you can, yes, I wanna do that. So I think I don't, except that I may at the very end just wanna put up like a closing slide.
Great,
Okay, so.
sounds good.
Oops.
I think we got a lot of questions here from the audience that we can cover.
Awesome.
Okay.
Questions are the best part, so.
Yes, that is the best part. All right, the next question is, how do you know if the antibiotic will work on a person? Do you have to test it out on a group? Like, how does it go from a thing that you found in the soil that makes it to a pill?
Yeah, so that's one of the things I actually have my students do in the classroom, and one of the things we talk about a lot is the specificity of an antibiotic, because so what we're really looking for are microbes that are producing metabolites, and some of those metabolites or some of those molecules have activity against bacteria. But that doesn't mean they don't have bacteria against other things. And so ways that I actually have students set up experiments in the lab, and they get to design their entire experiments is testing on things like fruit flies or on some fungi that we have that are a good model, because fungi are eukaryotic cells, so they're more like our human cells. And it's the first step of being able to say, do these things produce something that only has activity against bacteria or has more broader activity? And those can be important molecules because we need things that, for example, can have impacts on eukaryotic cells, because that's how we find things like cancer treatments or antibiotics that have activity against fungal pathogens, which are also something that are a growing concern. Totally. But that's what we do. So then you might use human cell lines. So test them first against human cells that we have that we can grow in the lab, and we can see that whether or not they have activity against that. And then they move into other animal models. So if you have a candidate that looks really promising in terms of its activity, if it looks new, and we can figure that out more molecularly. We can kind of, there's databases where we can look at the different molecules in that mixture and say, is this something new, or is this something we know about? Or, oh, is this something, that really looks new and it has really interesting activity, and this we want to prioritize. And those are the things that then would then be moved into these future steps. But eventually they go into clinical trials, and they get tested in humans to see if they work, if people have allergic reactions to them or things like that, which are important to know. So some of you may, you yourself or have family members who have a penicillin allergy is one that are sulfa drugs that gets talked about a lot. And so these are the important things we have to figure out before they can get used on humans. And the other important thing to consider, though, is not all of our important antibiotics are ones that we take orally. So there can be, they can have activity, they can be problematic to take by mouth, but they might be perfectly fine to put on your skin. So that if you have like a staph infection on your skin, you could treat your skin, and because you're not ingesting it, it's not necessarily a problem. And so these are things that people do as they're trying to study these, and that's why it takes a while, which is this concern where we're thinking out to 2050 is that might seem like a long time, but it's going to take us, something that a student finds, for example, in class next week, it's years to be able to get that to a point where we can use it.
Awesome. Thank you for answering that. That's really cool. Kelly, age 8, wants to know, what happens if you're allergic to penicillin?
So, I mean, so people can have problems where it will cause like an allergic reaction that can be very harmful. Other times it can be things where you just have like itchy reactions and it's just unpleasant. Typically, that's what happens is people they just feel unwell. They might break out with a rash or something like that. And what happens is that then that, you need to put that in your medical record and then make sure that if you ever need an antibiotic that you can avoid that type of antibiotic, because antibiotics come in different flavors and we call them different classes of molecules. And so it's easy to then, if you're allergic to penicillin, to ignore that and say, okay, we can try these other things. It becomes less easy as we get to having more and more resistance, but we do still have a pretty decent, toolkit for being able to avoid those things so that you wouldn't be having to take it. But that's why it's important to, if you notice anything weird after taking any sort of drug, to be able to understand and know that, okay, I should not take that again.
Right, for sure.
And talk to your physician about it. Yep.
Yeah. All right, we've got a question that I don't understand, but you may. So you might need to explain to us what this question means. But Khalid wants to know, have you found ESBL producers?
ESBL producers. Khalid, can you
we'll just say we'll skip that one. Khalid, if you want to give us an explanation of what that is, we'll try to ask it again. But we'll go on to the next one for now, because a lot of these I know you definitely will know the answer to.
Are you meaning extended spectrum beta lactamases?
I suppose so. Yeah, that makes sense.
At this point, I don't know. I mean, we're testing the types of microbes that could produce those things, and we're testing against microbes that could be sensitive to them. But I wouldn't be able to completely answer that in terms of whether someone has identified something like that.
Cool. Patricia wants to know, is there a place where I can find detailed instructions for students to do this project?
So I have a final slide that tells you exactly how to join us. We have a book that also lays out the protocols and things like that. We train, so I myself have trained about 200 instructors to do this. We're doing virtual trainings right now, obviously, because of everything.
Tiny Earth Network, right?
Tiny Earth, yeah. And so if you search Tiny Earth on Google, I think it's tinyearth.wisc.edu, you can find our site and there's information there.
Awesome. Madison would like to know, is there an antibiotic for cancer?
Well, they're not really antibiotics per se. They're molecules that microbes produce. So there actually are some cancer treatments that come from, there are some really well-known cases where they've come from microbes that live in invertebrates that live in the ocean. So some bryostatins and things like that are very effective cancer agents. What I would say is that what we call these when they're produced by microbes especially is metabolites, or especially you might see it written as secondary metabolites, because they're not the main things we think about. They're sort of extra molecules they produce that aren't necessary for sort of like everyday life, but that are important that have these additional functions. That's not necessarily the best explanation, but so they're metabolites. They're molecules that these organisms produce, and so there definitely are some that are so broad that they can have activity against both bacteria and eukaryotes, which we are. And so because of that, you can imagine that they could have applications in this at the right, especially depending on the concentration you use them at.
Totally. Awesome. Dean would like to know, have you found an antibiotic that can stop the flu?
So that's a great question, because it's really important in the context of what this week is about, which is also antibiotic stewardship. So antibiotics are not effective against viruses. And so that's why one of the biggest things we can do, even more so than trying to find new ones as sort of everyday people, is make sure we're using them smartly. So you should not take antibiotics if you have the flu, because it won't have an impact on viruses. The types of cell structures that antibiotics function against, we don't have in viruses. And so what you actually can do if you do have a flu or a cold that's also caused by a virus and take antibiotics is that you can cause other microbes that are on your body, bacteria, to become resistant. And so that's why we really try to get people to be more smart about their use, or to pay more attention to how they're using them. Now, it is true that sometimes when people have, like with what you may have read about different things like with COVID, where it's causing so much congestion in the lungs that people can develop a bacterial pneumonia on top of their viral infection. And then we do have to treat with antibiotics. But to treat for a virus specifically, it won't work. And so that's why it's really important that if you are just having a common cold or you have the flu, that you don't go to your doctor and try to get a prescription for an antibiotic.
Awesome. Well, thank you for asking that question, because
it's a super important question. Yeah.
Could you explain to us generally how antibiotic resistance happens in the first place?
Yes. So the question about growth is one of the biggest reasons. Bacteria grow a lot faster than we do, and they grow what we call exponentially. So one cell doubles and splits into two, and then those two split and you get four, and so on and so on. So it grows in powers of two, if you think about it that way. So you can go from one cell to 1 million cells in a very short period of time. What happens is as bacteria do that, they naturally acquire mutations. So they have a slight change, and this might change the DNA a bit, or it might cause them to suddenly produce, their cell structure to be slightly different, or to produce something that can actually deactivate an antibiotic. And those are in a population, even if it's 1 in 1 million, normally you're not going to see anything. But in the presence of an antibiotic, that one survives. And then it suddenly becomes 1 million itself in a very short period of time. And so that's the big issue, is that the bacteria so all life on Earth, when our DNA makes copies of itself, it's not 100% perfect. It makes mistakes. So we always have mutations that happen. Typically, mutation, you won't see the impact of it. But in the case of when there's suddenly antibiotic pressure, that resistant microbe becomes the thing that survives, and then it becomes dominant in the population. What's really interesting is actually, in a lot of cases, microbes that are resistant are wimpier. There's a cost associated with having that resistance. And so if you stop using the antibiotic, they'll actually become less abundant in the population, because they sometimes don't grow as fast. It takes them longer to, they need more energy to be able to do these things that make them resistant. So if there's no antibiotic pressure, if there's not the selection, they actually will become less abundant. And so in some cases, what some people have proposed in certain areas is if we stop using the antibiotic, usually in a period of time, we can actually go back to using it. But the biggest problem we have is every time we start using an antibiotic, we see resistance become present in the population of microbes in a very short period of time. But yeah, so it's that bacteria grow fast. We all have mutations. As long as there's selection, you select for resistant bacteria, and then they become the dominant in the population.
Right, awesome. Aubrey and Zadie would like to know, how long does it take to develop an antibiotic?
So I should know this number more specifically, but I think in most cases, the numbers that are given is it can take almost 10 years for it to go from initial discovery into where it's being produced in where you can get a prescription, go to the pharmacy and get it. It takes a while.
Sounds good.
It's years at least as a way to think about it. And it can be millions of dollars to be able to do that as well. So it's expensive too.
Cool. Let's see. So a lot of questions here. So does each antibiotic that we develop, does it act on bacteria in its own way? Or what is the difference between different types of antibiotics?
So they target a lot of different cell functions differently, which is why having these what we call different classes is really important. So some will target the cell wall of bacteria. So bacteria produce a specific molecule called peptidoglycan, which makes their cell walls rigid and helps give them structure. And it's a great target because human cells don't have it. Other animal cells don't have it. And so that's like the ideal target, something that's in a microbe, but not in a human or an animal cell, right? But in a lot of cases, that's a limited number. So that's what actually, vancomycin, if you've heard about that, that's affecting peptidoglycan. Then they affect other things. They can affect how DNA replicates. They can affect how DNA is transcribed into messenger RNA, which is what we need for producing genes. Or they can even affect then how that becomes proteins. They can affect metabolite pathways. And so we have some examples of really good antibiotics that because it's a synthetic pathway that a microbe has and a human doesn't have. It's a really great target. But we can also play with that in different ways. So they can, in some cases, things like DNA replication is actually really similar across all of life. But there's enough differences that we can target the ones that are specific to bacteria or have greater specificity to bacteria and be able to avoid those that have activity against microbes.
Sounds good. So let's see. What kinds of diseases do you mostly want to go to the doctor to get antibiotics for?
So if you have, things like staph infections, if you have, so streptococcus can be a problem for there's a controversy about whether for ear infections you want to use it, but there are definitely things like if you're having, what's the thing with the sore throat?
Strep throat.
Strep throat, yes, streptococcus. Things like that can be really problematic. If you have any sort of infection that's producing, like a bad pus or things like that, you want to, probably see a doctor about that and have like a topical antibiotic. And then there's other things that we know are problematic. So things that can become chronic infections. So Microbiome tuberculosis, if people get that and it's in their lungs, that's something that needs to be treated with an antibiotic. So I think, the biggest ones to think, if you're ever concerned, it's always better to go to a doctor and get checked out. But if all of your symptoms are just like the flu, then that's a cold, no antibiotics, but other things, it's always good to find out from your doctor. Certainly really bad inflammation that's throbbing, that's hot, anything like that, you should definitely get checked out.
For sure. Awesome, thanks. Gage would like to know, how do antibiotics affect your beneficial bacteria that live on our skin and in our gut? He uses the word gut flora, but we know there are lots of different types of bacteria all over us that are super helpful.
Yeah, so that's actually what my lab mostly studies. We study the gut flora or microbiome or microbiota, you may have heard it called. And it's something that we know and I think have a growing appreciation for, the number of ways that it keeps us healthy and is really important. And so we know that antibiotics definitely can affect our microbiome. Typically that means if we're taking an antibiotic, it's because it's more important to treat the infection than the temporary impact it will have on our microbiome. Because what people have been able to show in a lot of cases is that, especially if you are 2 years or older, where your microbiome is sort of established what it's going to look like for the rest of your life, the exposure to your antibiotic might reduce your numbers or change a bit the composition, but most healthy adults will rebound and it will repopulate normally. It can be a problem if it's more of an immature microbiome. So in young children, there's concern about that. And so that's another reason why we say, it's important not to take them if you don't need them, because it can both affect the composition and the number of microbiome members, but it also can mean that you have microbes on you that are also becoming more resistant to antibiotics, which in the future makes them, even if they're not causing illness to you, it means that the antibiotics we have will be less effective.
Very cool. Jesse would like to know, why is it important to take antibiotics exactly as prescribed?
Yeah, so a lot of times what you might see is that you're supposed to take it a certain number of times a day and for so many days. So the certain numbers of times a day actually gets a bit at the bacterial growth. So if it's a pathogen is growing, so when they're growing, they're more sensitive. So you might need to, just taking one dose isn't necessarily going to be enough for the antibiotic to be there present while more pathogens can grow. So we take it at certain times so that you can get another dose, knock back the populations more that might have survived the first dose. And that's also for the number of days why we do that. We want to make sure that you're not reducing and getting rid of most of the bacteria, but there's still some there, that we really want to make sure that if there's an infection where there's bacteria present, that we're really getting rid of all of that there.
Awesome, for sure. That can be super dangerous. Amara wants to know, so you find a bacterium that is really good at killing something else, and then you send it to the chemistry lab, you pull out this chemical, this metabolite that is the thing that kills that other organism. Is it the same process to figure out if that one individual metabolite is super effective as the bacteria? Like, how do you know that that one thing is the thing that could be developed into a drug?
Yeah, so the way that that's done is initially looking at, so you first have a mixture of different molecules or chemicals in that initial extract that you're looking at. And then what you can do is that typically the way chemists or natural products chemists look at this is that based on their size and the structure of the molecules, they can differentiate them. And then they can start to separate out and test those specific molecular entities to identify which is the one that has the biggest impact. And sometimes they can produce many. You can have 3 different antibiotics produced by the same bacteria, but it could be that the one that is the most interesting to you is a single one, or it might be that actually the way they produce them together gives you sort of the impact that you want. But that's how it's typically done. So they kind of separate out by size, test the individual ones, and then look for which has the activity that's the most desirable.
Awesome. That sounds fun. Franklin wants to know, can bacteria lose their resistance to antibiotics?
So yes, they can. But you have to be careful how we think about it. So some resistance is based on pieces of mobile DNA that we call plasmids or even transposons. And so plasmids are a circular piece of DNA that bacteria can take up from the environment. And this is actually a really important way microbes share resistance genes with each other, because you don't have to be the same species of microbe to be able to share these in a lot of cases. And so if a bacteria would lose that plasmid, which can happen naturally or through some other selection event, then they could lose that resistance. That's actually what happens in the case where I said is if the selection pressure is there, they need to keep it, right? But it costs more energy to replicate that extra piece of DNA. And so if the selection isn't there, you're going to grow slower if you're that bacteria with that. So you might then want to lose it and get rid of it because you don't need it. And so that can happen. Other types of changes, it's not, there are other types of resistance mechanisms that aren't that easy to gain or lose. What happens instead is it's their abundance within the population. So you have to think about the population of different microbes. And so that's how that happens. But it can in the case of something like if the resistance gene is on mobile DNA.
Awesome. Well, we try to keep these sessions to be about 45 minutes, and we are coming up to that right now. This has been so awesome. We've had so many wonderful questions. Thank you all for submitting your questions today. And before we wrap up, we always ask everybody the same two questions. The first question is, if you had the attention of everyone in the world and you could tell them one thing about your area of expertise, what would that be?
All right, I'm going to do that by also going back to my slide I wanted to share, which is to say, you can have a role in this. You are an important part of making this happen, either through participating with something like scientific discovery through a program like Tiny Earth, but also by just the choices that you make and the information that you share with other people. So, you know, be mindful about antibiotics. Don't take them if you don't need them, and think about how they're being used in the environment around you, and make sure that you tell the people, you know, that you live with and that you interact with, that this is an important thing to pay attention to. And that by being aware of these things, you can make a difference. And so I am highlighting that, you know, we have World Antibiotic Awareness Week. The CDC has a really great campaign about this, about be smart about antibiotic use. We also have things like World Soil Day coming up, and you can learn more about and actually participate in things that also help educate about the importance of things like our soils and why keeping them healthy is a really big part of us being able to discover new things like this. So that's, I think, one of the important things that I would have you take away from this.
Awesome. That sounds great. And then we also tend to ask if you still have everybody's attention in the world and you can tell them one thing that has nothing to do with your area of expertise, just something. It can be as big picture important or silly and insignificant as you'd like.
Well, I'm gonna keep on a microbial thing. So I'm gonna say what we talked about is ways microbes can be problems, right? So they can be pathogens, and we have this concern about antibiotic resistance, but they're part of the solution in this case, right? So the microbes are also where we can go and find antibiotic production. And most of the microbes that we interact with are beneficial for us. They help us, you know, they make oxygen for the earth. They help us with our own physiology as animals, with our microbiome. So don't be afraid of bacteria. Be smart about how we think about them, but microbes are also fantastic things, and you should embrace them. And my favorite microbe is Bacillus thuringiensis.
That's awesome. Thank you so much. All right. Well, thank you for sharing that. Where can we find you on social media? Do you have anything else that you'd like to plug before we wrap up?
So you can find Tiny Earth. Just search for us at Tiny Earth net. And we're on Instagram, TikTok. We have a lot of stuff going on for World Antibiotic Awareness Week. So you can see some of the really creative things our students do in addition to just making awesome discoveries. And if you're interested in this, we have a website where you can either ask questions or you can fill out an application to join us. We'll be doing virtual trainings for the near future, but looking forward to getting back to in-person trainings when it's safe.
Awesome. Well, thank you for taking the time to talk with us today. Erin, thank you for signing.
Yes, thank you, Erin.
Everyone, thank you for joining us. We will be back here on December 3rd. We're going to be talking with the extremely energetic, extremely animated and entertaining and funny Jada Elcock. She'll be talking to us about marine science broadly. She is an expert in sharks. She's just, I can't overemphasize.
She's awesome. She's fantastic.
So get excited. That's going to be part of Black and Marine Science Week. And we're pumped. We can't wait. So get excited for that. Mark your calendars. You can always keep up with what we're doing at skypeascientist.com. We also are trying to build our Patreon support right now. So if you can afford to, you can support us for as little as $3 a month on Skype or on patreon.com/SkypeAScientist. And right now we're doing a little promotion where if you sign up as a new patron, we'll send you a little, not necessarily this specific postcard, but we'll send you a cute animal postcard in the mail with a squid fact on the back. And we'll send that out this week. Whenever you sign up, we'll send it out same day. So if you'll do that, that would be so important to us to keep us going in strong into 2021. So that's patreon.com/SkypeAScientist. We'll see you all back here in December. Have a healthy holiday season. We'll see you soon. Thank you again, Nichole and Erin. Bye.
Thank you.